research that goes on at the Ski Institute. I'll start with kind of an overview of what is this whole Ski Institute thing, and feel free to interrupt with questions anytime. I'll try and be as concrete as I can too, about opportunities for research that exist and how you can tie into some of those. And because you probably believe your fellow students better than you believe me, a lot of the talks we're going to have coming afterwards about the research are from grad students who work here at the Ski Institute, and can tell you from their perspective as students how they got involved, what kind of research they actually perform. And some of them started out as undergrads. This is a pretty common pattern that we have students who come to us as freshmen in some cases. A number of them have come from this program, in fact, and got involved in research here, stayed working with the Ski Institute throughout their undergraduate careers. Some of them stayed as grad students, some have gone on to other places and bigger and better things, but there's a long history of undergrads making their way through this place and finding this is a really good and productive home for their research. And part of the attraction, I think, of the place is the broad variety of the topics and subjects of research that we do here. So I'll talk a little more about the faculty in a bit, but the faculty are very broadly spread across the campus. I'm not a computer scientist at all, and I'm trained in physiology. And we have other colleagues here who are also in bioengineering, other colleagues in electrical engineering, so we come from a variety of disciplines. And what brings us together, what really ties us all together, makes us collaborate as much as we do, is this notion of computing, and the use of computing and computers and a lot of, not everything that goes with them, but a lot of different uses of computers in a number of application domains. And so we'll try and hit on some of those application domains. You get a sense of what that's about. But the theme is not computer science or bioengineering or any one topic domain. It's really the use of computing and modern computing techniques applied to some discipline. So if you want to learn about how compilers work and the architecture of computing, that's not really what SCI does. Computer science does that. If you want to learn about the specifics of biochemistry and how the Krebs cycle works and how molecular biology works, this is also not the best place for you to be. But if you want to understand how computing and science come together in an incredibly broad variety of topics, SCI is the best place to be. And not just the best on the campus, I think we're one of the best places nationally and even internationally, and we're known for that nationally and internationally, as being this interdisciplinary place with a real range of topics. So already here you see a whole bunch of pretty pictures. We obviously do lots of that. You've seen some of the posters outside. We have a reputation for our pretty pictures. But there's more, of course, behind each one of these pretty pictures, and we'll cover some of that as we go along. Now, what some of you may not know, unless you came to last week's SCIX, how many did that? Anyone know about SCIX or come to that last week? Okay, so we had a big open house here last week on Thursday for internal consumption, Friday for the public, and this man right here came. Anyone know who that is? Anyone recognize him? John Warnick, exactly. You're in the building that he helped create. So he is the major benefactor, and you're the major beneficiaries of this building. So John Warnick was a graduate of computer science here at the University of Utah, went on from Utah to work for Xerox PARC, which was you could argue, the home of personal computing. This is where the mouse was developed. This is where graphical user interfaces were developed. Steve Jobs dropped by Xerox PARC in California and got a tour of the place and just stole all the ideas he could and took those ideas and did the really impressive thing, and that's turned them into practice, made actual software and hardware to support those ideas. They were just sort of pie in the sky type things back then at Xerox PARC. And so John Warnick went through Xerox PARC and then created Adobe, that rather small company that makes insignificant software like Photoshop. So John Warnick is now a very famous guy, a very wealthy guy too, as you can imagine, and he's an alum of the University of Utah in computer science, but he's not the only one. Here are some other people. This is David Evans, and he went on, he was actually the founder of the computer science department here, went on to create a company called Evans and Sutherland, and they were really the company that first started doing interactive simulations. So you've all heard of flight simulators, right? You probably played games, flight simulator games. You can do that on your iPad these days. But back in the day, that was a huge endeavor to have realistic, real-time simulation software that would allow people to steer and get responsive feedback from that steering. And Evans and Sutherland was really the forefront of that. Most of their simulations ended up in the military domains. Their first tank simulators and flight simulators used for military purposes came out of Evans and Sutherland. Who else is here that you might remember? This guy here is Jim Clark. So he founded, again, a company called SCI that you probably haven't heard of, but SCI really developed arguably the first effective, efficient graphics hardware that you now enjoy in all of your devices that you see around you. And out of that evolved a standard language for describing graphics, and companies like NVIDIA that make those super graphics cards you all enjoy playing games on, that really came out of the SGI effort. So that really started with, again, Jim Clark, a graduate from the University of Utah. And there's other people here, again, depending on how deep you are into computer science, you may or may not know about. Ed Catmull is certainly somebody you might know about. That's an old picture of him. But Ed Catmull went on to, again, sort of form a small company called Pixar, which joined with another tiny company called Disney. And he heads that company and is responsible for some of the fantastic animations that we all go to pay a lot of money for to watch at the movie theater. So again, a long history going back quite a ways now to the University of Utah and the graphics and visualization. And that formed kind of an underpinning or motivation. It's not so much the topic area per se, but for the atmosphere that we wanted to create at SCI. The reason this all happened back then was the unique atmosphere, interactive, interdisciplinary setting that brought all these people together under one roof and said, just do great stuff. Don't worry too much about where exactly the money's going to come from. And don't worry, for heaven's sakes, for making a product, but do research. Take it into new directions. Take ideas that you have and pursue them, and we'll try and figure out how to support that. And that motive, that motivation, is really what still drives SCI today. So getting involved with SCI gives you an opportunity to dig into lots of existing projects, but also the opportunity to pursue new and crazy ideas and find people who will actually listen to you. So that's what SCI's about. Now, if you want to boil it down to some topic areas that we're engaged in, there's three main ones, you could argue. Visualization, you've seen examples of that, again, goes back directly to that early graphics history of Utah. Computing is another one. So computing, we see under that this sort of simulation of reality, the capturing of biological or physical processes, and translating those into a computer simulation so that we can simulate a heartbeat or the electrical signals from the brain or fire. And do that in a way that's relatively realistic and allows us to explore some of the parameters involved in that physics or biology. And then image analysis, that's the third big driver. And so images are all around us. Images are what we capture in our cameras, and they're what obviously we capture at the hospital, through MRI and CT and all the various modalities that are really changing the face of medicine. And so the analysis of that type of image information and extraction of function and structure from that image information is another one of the big three topics here. So most of us sort of fit somewhere in that domain, and most of us fit in all three. That's the key thing about SCI. So it's not a compartmentalized place, it's a highly integrated place. And you'll see that with some of the topics that students will give you. Now, out of each one of those three areas of expertise and areas of interest have come software products. So the other thing we do at SCI is we develop software. We don't just develop it as internal use only, something that we hack together to solve a particular problem. We go to the next step, and that's we make software that we can give away at least to people, and that people are interested enough in to download and use for their applications. So you can go to our website and download any of more than a dozen different software products that have all been developed here at SCI as part of funded projects to solve particular problems that we then generalize and made useful for the broader community. And so you may come across a topic here that interests you. You may come across, through your own existing involvement with other labs here, topics and research and software that could support that endeavor. And again, you're free to download and use that software. So out of these three areas, in each domain has come software, and that's really what we do. So here's some examples of some of the things we do. This is obviously a rendering of a geometry of a skeleton. You can see the bone here, but you can see also the bloodstream here. And here are the kidneys, and I don't know if you can't see the heart very well here, but we extract information from image data sets at high resolution. We manipulate those and visualize those in interactive ways. And so these are all kind of examples taken from the human body using different tools that we've developed to identify structural information from the image data sets. Here's again, that's obviously a torso, a piece of a thorax. This is the kind of structures that we build, patient-specific models that we build in which to carry out simulations. And one of the students, Jess, will explain how we use that to, for example, simulate defibrillation, simulate the application of electric potentials to save somebody's heart, to save somebody's life when their heart goes into abnormal rhythms. And here's some other things that are totally far afield. This is the plasma field that fusion scientists, physicists create in order to try and create the setting, the situation that will support nuclear fusion research. So we don't do fusion research here in Utah. We reach out to, or other researchers in physics departments in other national labs reach out to us to get the support for the visualization of some of these enormous data sets that they produce from their simulations. This is a hand. So we look at things like structure extracted from images. We look at the variation, the statistical variation of anatomy, of structural changes across individual animal species examples, across individual humans. We have a study ongoing now where we're sampling the shape of left atria from patients with a disease called atrial fibrillation and trying to characterize the shape of those left atria, because it turns out AFib is a left atrial disease, and see if the variation in that shape correlates to the variation in the extent of the disease that these patients have. And so shape and the extraction of shape and characterization of shape statistically is another topic of big interest. Here's an interesting example. This is a cross-section through a nerve bundle. I'll come back to this image of one like it in just a sec. So we're interested in following the nerve bundles, in this case the optic nerve, and trying to reconstruct how the neurons connect across slices to build a three-dimensional map of what this nerve bundle looks like and how the nerves start in the eye and go to the brain and how the processing and the interactions of those nerves can actually function. This one here is an explosion. This is a rapid fire building up of pressure inside a chamber, and that chamber eventually exploded. So there's a huge variety of topics here. We've spun off companies. We're involved in a lot of research, but we also spin off companies. This is one of the ones that NVIDIA has purchased, where the head of this company is the leader of an NVIDIA research group here in Utah, Special Center of Excellence. This is the center of excellence that they sort of gave us in return. We have a pile of hardware upstairs. We do large-scale simulations using the graphical processing units that, on the one hand, are what make graphics cards go quicker, but on the other hand, have now become general-purpose computing structures. They become a new form of computing. And so we do research at this sort of high-end, high-performance computing using this GPU cluster, which looks like this, but is really impressive because the accelerations it can make possible in analyzing large-scale data like this. These are MRI slices of human heads at various stages of development. It takes enormous processing power to extract the shape information, the structural information from large data sets like this. And we combine these sort of biological problems of large scale with the rapid and large-scale computing and develop the research tools to make that possible. So we work between this application domain and the computing domain. Namira Biosciences is another local company that lives over in Research Park that's spun off out of SCI. They have imaging modalities. They create contrast agent dyes that they can use in what's called micro-CT. So this is computed tomography down to the scale of microns. They take tissue samples that are less than a centimeter cubed, and they're able to scan those at resolutions below 10 microns. Now we're getting down to the size of cells, individual cells inside biological tissues. And so they do virtual histology. And we work with them to do visualizations. This is actually the skeletal structure of a mouse embryo of a transgenic mouse. So this is a mouse whose genes have been altered to alter the shape and the skeletal development of that mouse. And from the embryo of that mouse, which you can imagine is about this big, they can do this sort of high-resolution imaging, and we provide some of the software tools for rendering and display of those data sets. Vistrails is another company that is, again, spun out of SCI, and their business is really keeping track of interactive software decisions that we all make. So when you sit down with a Photoshop application, for example, and start tweaking the photos, and you play with the color table, and you crop it, and you scale it, and you make substitutions for colors, all those steps are lost. Right? 3 months after you've made a nice image, you can't possibly reproduce that image. You have no idea anymore exactly which parameters you used, which steps you took to create it. That's fine when it's your own photo, you don't really care. It's almost a disaster in science when at the end of a process you produce a result that you can never replicate. Right? You all know enough about science. You know that replication is one of the key steps in the scientific method. Right? If you can't replicate it, it hasn't happened. It doesn't exist. So the idea of Vistrails is to capture what's called provenance information, to capture a list and do it automatically of all the steps and decisions that you make as you're processing data. So it's a very generic tool that we're applying to a lot of different settings, working together with this company. Now, we're also the home of a number of national research centers. So SCI is the home of things like the NCRR funded, this is an NIH institute called Center for Integrative Biomedical Computing, called, of course, the CIBC. So here we, again, bring computing tools together in an integrative way, merge image processing tools with simulation tools and visualization tools to solve problems that are of interest in biomedicine. And we have NIH funding to do that. We've had this grant since 1999. We just got it renewed last year for another 5 years, and just got another supplement added this year. So it's been a very successful project, and kind of typical of the kind of projects that are really well suited to the Ski Institute. We're also affiliated with other centers. So NAMIC, for example, is another national NIH-funded center that's based on the notion that imaging, computing from imaging, and extracting information from images is a broadly applicable domain, and that there's a driving need for software and technology to perform those tasks. And so we provide some of our approaches to their software and work together with them. Here's just an overview of where our money comes from. So we do an enormous amount of research here at SCI. NSF provides a chunk of our money. NIH provides the dominant chunk of our money. We also have support directly from industry and ongoing support from things like the Department of Energy. They're interested in high-performance computing as well. This pie chart varies from year to year. This is the pie chart for this year. It shifts around a little bit, but NIH and biomedical research is a big part of it, but so is fundamental research coming from sources like the NSF. Now we have a few connections here among our faculty. So these are the full face shots of different faculties, the different groups within SCI, and these are just a summary of some of the external collaborations. So we work not just with researchers here at the University of Utah, but with researchers in national labs and universities around the country and around the world. So SCI is really a nexus, is really a hub for a whole field of research collaborations that truly go far afield. And as you can see, touch leading institutions all over the country and the world. And again, being part of SCI, you can be part of that. We do have sort of an overall vision of how we see the kind of software environment of the future. And here we're going to apply it to some biomedical situations, but you can imagine it's applied to other settings. And that is that we see that there's a loop, there's a feedback process that we're trying to achieve through software that starts with acquiring images. You see in the upper left here, acquiring, in this case, image data from patients, let's say, using MRI, processing those images, extracting geometric information. This is anatomical information. Geometry allows us to build models. With those models, we can carry out modeling, simulation, visualization. You'll hear some examples of that. This is the simulation of the electric field applied to a metal implant in the residual limb of an amputee. Somebody came to us from orthopedics and said, I think we can do a better job with amputees than the socket interface they have now. Right? They have a stump left after they've unfortunately lost a limb, and they have a socket of a prosthetic device that fits over that. This turns out to be a horrible interface. Friction is an ongoing problem. Skin wear, skin lesions are problems. Infections are problems. And so they've come up with an idea of inserting a metal rod into the remnant bone, and letting that rod grow into that bone, or the bone grow into the rod, better said, and then using that rod as an attachment for prosthetic devices. And when this works, it's phenomenal. The patients, the subjects who experience this successfully have greatly expanded mobility, and their ability to live a normal life is dramatically enhanced. So they came to us and said, yeah, we need some software to help simulate how the electric fields might arise, if we could accelerate the bone growth by applying electric fields. And so we developed our software, modified our software for that application. Visualization is a key part of everything we do. Again, you've seen lots of pretty pictures, you see lots more here. But we really wanted to take all this and feed it back into the lab, into the clinical domain, taking that image information, all the steps we extract, and feeding it back to the clinician. And to that end, we do work with clinicians. Here's an example of a project you'll hear more about, in which we simulate defibrillation. That's a tool that physicians can use to simulate the placement of an implantable cardiac defibrillator in a patient before they actually undergo the operation, to try and optimize the placement of that device, to minimize the shock strength, minimize battery use, improve outcomes. Here's an example of a physician here from the University of Utah. This is Nasir Marouche. He's a cardiac electrophysiologist. He sticks catheters inside people who have abnormal heart rhythms and tries to cure them by burning, by ablating the abnormal tissue. And here's a system where we're actually taking information acquired from the MRI magnet. He's got his arm stuck inside a 3 Tesla MRI magnet here on the campus. We're taking the signals from that, feeding it through a computer, driving it back onto the screen, and he's seeing right here a projection of the images and the information we extract from those images back onto a display to help him steer the catheter to put it in the right place and carry out the operations that he does more efficiently, more effectively. So here's an example of that loop being closed in the clinical domain. We also have had the pleasure of working with Mara Kopecky, who you may know is the only Nobel Prize winner from the University of Utah. In that domain, we helped with, again, visualization, functional anatomy measurements from, in this case again, these transgenically modified mice. And that's an ongoing project with Mario, and especially with his then student and now independent researcher, Charles Keller. Here's an example I showed you before of a slice of the neuron. And this just looks like, you know, a regular slice. You might, you know, if I made you guess, you might guess, you know, there's sort of some number of pixels, I don't know, 1,000 or so pixels across, and, you know, it's some kind of image. You'd be dramatically mistaken, because what this is really a mosaic of multiple individual micrographs, so microscopic images, all pulled together. You know how you can buy software to sew together those panorama shots you do with your camera, right? When you stand on the Grand Canyon, take a bunch of photos. This is the same idea, but on a grand scale, because this is actually a three-dimensional data set. It has 341 sections. So this is just one slice, so 341 sections. Each one of these sections is 90 nanometers, so this is very, very fine scale. Each one of these sections has 1,000 individual tiles, roughly. So each one of these pictures here, these little blocks here, that's one image. Each one of those is 4,000 by 4,000 pixels. So that's already huge compared to computer screen resolution. And there's 1,000 of those in this image here you're seeing. So it's 1,000 times 4,000 by 4,000 pixels. That ends up being 32 gigabytes of data, that one section. And there are 341 of those sections. So at the end of the day, this is 16.5 terabytes. 1,000 gigabytes of data. And researchers bring that to us and say, help! What can I do with this data? I can't even store it on my disk, you know, and I want to see it, I want to visualize it, I want to find the nerve bundles going through this data set. And so that's where we help them. We help them with merging all these images, bringing it all together. We help them with tools to analyze, to follow the neurons, just to look at these data sets is already a challenge in itself. And so that's the scale of things that we do here at the University of Utah. We make tools, computer tools that span a bunch of different platforms. Here's an iPhone application you can download right now. It's called ImageViz 3D. And ImageViz 3D on a desktop is capable of handling these huge data sets and allowing us to visualize those interactively. You have to go a little smaller on the data sets to get it onto your iPhone, but you can still take visual volume data sets and visualize them in real time, manipulate them in real time, move them around, slice them, dice them, see different parts of the skeletal structure as you see here in this image, in something as small as an iPhone app. So how do you now get involved? So what's your access point to this whole great world of amazing computing? It's really through these folks. So these are the faculty here at the SCI Institute. We all have different skills. We all come from different departments. Some of us from multiple departments. You know, I'm in bioengineering and cardiology. It's my appointment span. And that's typical of a lot of the people in the department with multiple appointments across the entire campus. And so these are the people you need to contact, and these are the people you can track down from our website. Find out what their research interests are, and see if they match some of the things that you want to do, you might be interested in engaging with. And if you are interested, contact these people. Send an email. Say, I'm an undergrad student really interested in research, and I read about your paper or your topic on such, and I'd love to have a little bit of your time to find out if there's an opportunity to work in your lab. And because we're all insanely busy people, pester us. Don't give up the first time. And that's what I tell all the I'm the director of the undergrad program of bioengineering. The first thing I say to all the students is, you know, be persistent in this game. Be aggressive, be persistent, and chase people down who you think can help. That's the way the world works. So these are the people you need to check out and get to know and find out more about them so that, you know, if you like, if the fit is right for you, you can be part of this amazing team that is now over 200 people here at the SCI Institute, a huge chunk of which are students. There's only 16 of us faculty. And there's a few staff people, and there's some developers, and an enormous number of students at all levels. And if you go through the website, SCI website, you'll find lots and lots of photographs of people who are students. You'll find a whole list of undergrad students who are already engaged in research here. You may even find some of your friends there, and you can contact them and ask. So with that, I think I'll finish up with my sort of overview presentation. Are there any questions at this point about SCI? Feel free during the rest of the presentation to pick up on that. And I've forgotten the schedule, so I'm not sure who's going to be talking next. So
Josh Blower is next.
All right, Josh Blower is next. So does Josh have slides? So Josh is a grad student of mine. He's a PhD student. He's involved in this project that I mentioned before about atrial fibrillation and that project with Nasir Marouche, a clinician. Josh's background is in bioengineering, and he's involved heavily in some of the image analysis that goes on. I think he's going to talk a little bit about bioelectrical fields in general. And that's a broad area of expertise that involves both the heart and the brain. Because it turns out, as you probably know, our hearts generate electricity, our brains generate electricity. And that electricity can be tied back to the function of those organs. And so sampling that electricity, understanding where it comes from, how to recover it, let's say from the body surface, are all valuable clinical approaches ultimately to help physicians better identify abnormalities and better treat them. And so this domain of bioelectricity is one that we pursue in a number of different settings. And so, yeah, hit the play button and see if that scales properly.
Oh.
That's the right guy.
That'll help.
There you go.
Good. Mind if I steal your, oh, it's that's plugged into your laptop. Never mind.
You can probably plug it into the other computer. Plug it in to whatever computer's driving the slides.
Is that even with you? And then there's James, who makes everything work. Well, I'll just get going, and if this comes to life, I'll give it a go. So yeah, glad I could be here today. As Rob mentioned, I'm a graduate student in bioengineering. I came through the undergraduate program here through the College of Engineering as a bioengineer. And so Rob mentioned kind of the workflow that we have, the whole idea behind software development at SCI, where it starts from acquiring whatever your data is, and then it moves through a path that hopefully circles back to actually be used in the real world. And so I'm going to show just a quick demo here of some of my work that illustrates how we can acquire this data, and then it can help us cycle back and to apply our solutions and our findings in the real world. So, as Rob was also mentioning, I work on what we call the AFib project. So I study atrial fibrillation. It's an arrhythmia of the heart. That's just where the upper chambers of the heart go out of their normal rhythm. So instead of getting your lub dub, it's just white noise. It's not doing anything it's supposed to be doing. And so we have a couple different things that we do with to study this disease. And the first is we acquire MRI images of patients who have atrial fibrillation. And so we can use these images to study different aspects of the disease. But to do that, we need to extract that information from those images. And so there's a process called segmentation. And segmentation allows us to isolate the particular information of interest from those images. So here for this particular case, I'm using software that's developed by the CIBC Center that Rob mentioned. And this allows me to go in and extract the geometry of interest from these MRIs. So this is the heart. This red chamber is the right atrium. That's the aorta. Stuff you may be familiar with. And once we get that information from our segmentation, we can build geometric models of it. So that can be used for visualization or further data analysis. I'll show a couple different examples. So one would be where we just build a model like this. So I've taken that right atrium, I've made a model just like you would kind of like with sticks. I can't remember exactly what they're called, like Tinker toys or whatever, where you can build a model from it. This is just a frame that represents the geometry of a right atrium. Now I can take that, and we have a project where we're working on guiding procedures that doctors actually perform in patients. So these procedures, they actually will stick something into somebody's heart, they'll guide it up through their veins, into their heart, and they need to know where these devices are. And so what we're trying to do is use MRI to guide the placement of it. So you'll see that this blue catheter, as we call it, this device, it'll come up into the heart, and they'll be able to position that up in part of the heart called of the atrium, called the appendage. And then they have this other device that they're also going to navigate up. And so using that model that I've built, so that's that red model that I showed you right before, that's integrated with the MRI so that it can guide the placement of devices in a subject. Another example would be, so again, we're using that same model, But now we're going around and we're using that catheter to go and record electrical signals from the heart. And so what these purple dots are, those are all the places that we've sampled electrical signals from the heart. Now we can take those electrical signals and we can use those to map information onto our models. So here what we've done is we've mapped activation time. So the heart, there's an electrical wave that passes through it, and the timing of that can tell you a lot about how healthy the heart is and how the heart is functioning. So in this model, what I've done is I've taken that parameter that we got from our electrical measurements, and I've mapped that back onto the image data, or the geometry that I created for the image data. And so this is just one more representation that we can give to physicians or people studying this disease so that they can understand how things are working in the heart. So here's another case where, again, we're doing that real-time MRI guidance, and now they're doing something about this problem of atrial fibrillation. They're going in and they're burning problem sites. And now when they've gone in and burned, we can see that with the MRI, and so we can show them, okay, you burned here and you burned here. And what this video shows is them using that information to help guide further treatments, so they can know where they've treated and they can use that to progress. And so this is real-time treatment, but we can also use it more in a diagnostic sense, where we can look at someone before they even go in for a procedure or after they've had a procedure to see, if it's before, we can see, is this procedure going to be successful? Do they meet the criteria necessary to get this procedure? Or after, we can say, how effective was this procedure? So here's a left atrium of a person a doctor has gone in and burns in their heart to treat atrial fibrillation. And this model shows in the red where they've gone in and burned. And so they can look for gaps where they were supposed to burn, and or places that they just completely missed. And so this is kind of that circle that Rob presented. This is more the visualization aspect, allowing the doctor to really go in and key in on what the image data is telling them. So this is an example of before ablation. So they can go in, here's the left atrium of the heart, and they'll go in and they'll segment that. And then using that segmentation, they can create models that show them where there's problem areas in the heart. So before they ever even stick that catheter into that person's heart, they can go in and assess where there's diseased tissue, so that they'll know once they go in there where they need to burn. And so the last thing that Rob mentioned is we can use all this data to simulate disease states, or we can understand and create simulations of, if we do this, what kind of result can we expect? And so here's some simulations that I've created where we take that image data and we say, okay, these green areas are the diseased tissue, and then we run a simulation of electrical activation of the heart, and we see how those diseased areas affect the activation of the heart. So you'll see this wavefront coming along, and you'll see how it circles around that area. Well, that's that green area. So we can start to get an understanding of how this diseased tissue affects normal function. And so these are all just kind of examples coming together of this image data, or just electrical data, all this data being combined using those basic set of software tools that Steve has developed to bring all the data together and analyze it in a way that becomes useful for scientists, for doctors, or for any range of professionals studying a wide variety of issues. For this example, I'm just showing how the orientation of that catheter with respect to the wall can affect the electrical signals. So this is something that doctors don't really think about when they're sticking that catheter up in somebody's heart. That, wait, where I put that catheter and how I put it there might affect my diagnosis of that particular tissue. So these are all just different aspects. And I'd be happy to answer any questions that you have about what I do. Yeah, please.
So is there any way for them to see if they burn too much?
So that is one of the driving, well, that's one of the biggest motivations for doing this real-time MRI that I was talking about. So frequently, well, as the procedure currently stands, they use X-ray to guide these catheters up into the heart. And so X-ray, they can kind of see a shadow of where the heart is, and they can see the catheter real well, because the catheter will block X-ray. But other than that, they don't have a good sense for how much they burnt. And so there can be some pretty serious complications if they burn too much or if they punch through the wall of the heart. I mean, death could easily result if they don't rush that person to surgery to fix the problem. And so what the benefit of MRI is that we can actually see what's going on in the tissue as they burn, so that they can look at it and say, okay, we can apply enough energy, but not so much that we're, damaging the patient or leaving them worse off than when they came in. Yeah.
How long have you been working on this project, and how long has this project been going?
So about 4 years for both. So I got on right about as this project started, and I've been here 4 years now.
Is that like the reason? Is that, and I guess going on with that, how much longer do you think until this project is completed, or is it just something that's going to just keep going and turn into something else?
So I've presented a wide variety of applications. Well, not wide, a couple examples of how we're using MRI. Some of these are used in clinic currently. So some of these, I wouldn't say it's complete, but some of them are tools that are being used every day up at the hospital. And we're one of the only centers in the world that is able to use this type of data in that aspect. But to say when it's complete, I mean, it's definitely ongoing. We're getting more and more information to help guide what the clinicians are doing. So in that 4 years, there are definitely things that are already being used in routine clinical care of these patients with atrial fibrillation, but it's not even anywhere near done. Yeah?
What do they use? This catheter can't be metal or have metal around it. If they're sticking it in an MRI, do you know what they use?
Good question. So, so an MRI creates a huge magnetic field. So if you go walking, there's videos you can find on YouTube. If you go strutting into an MRI room with an oxygen tank, that oxygen tank is going to go ballistic. And I mean, literally, it will become a ballistic object that'll fly into the bore and kill whoever's in between the bore of the magnet and the oxygen tank. So we can't lightly just walk in there with any sort of surgical instrument for these patients. So we work with a company, and I'm taking too much time, so this is probably my last question. But we work with a company that develops MRI compatible catheters. So when you say metal, what you can't use is ferromagnetic metals. So you can't take steel or anything of that nature. That actually is attracted to magnets. But you can use, say, gold, platinum, copper, and have it in the bore, and have it in the room with relative safety, although it can impact your image quality. So that's one of the huge challenges, is working in a magnetic field that big creates some unique problems that require a lot of innovative solutions, from mechanical engineers to electrical engineers to MRI physicists. I mean, we've got all types working on this project to try and overcome some of those challenges that you allude to.
Can I ask one more question? Can I ask one more? Just in your kind of day-to-day stuff, how much of what you do is code and how much of it is more like a design type interface for designing what we do?
Okay, so I am not a software developer, which is what the beauty of the CIBC software development program is. I can take these tools they make, and if worse comes to worse and it doesn't have the functionality that I need, they make it so it's pretty easy for someone like me to go in and extend that functionality to what I want to do. But I'm not writing the user interface or the core algorithms that the software is based on. So I don't know what your background may be, but for me, I'm not down in the nitty-gritty, but I'm kind of more on the end user side of the spectrum.
Okay, thanks, Scott.
Sorry for going long.
No, no problem. Very good. Okay, so the next speaker is going to be Defan Wan. He's a different perspective. He's a true computer scientist. So he understands math and computing in ways that Josh and all the other students don't. And he'll show you sort of another perspective on how you can apply some pretty advanced mathematical and computational techniques to a problem that's still very relevant to cardiology in this case.
Yes, so my background is computer science for my undergrads, and then I came here for the PhD, and where I switched to this more biomedical computing. So here I will show you how we can apply our math and the computer techniques to actually help build those facilities for real medical problems. So how many of you are familiar with so-called electrocardiography or ECG?
How familiar?
Yeah, do you, have you ever heard of this term, these words? Yes. So ECG essentially studies the electrical activity of the heart and try to make exploitation for clinical purposes. If you go to the hospital today for diagnosis, the standard practice is called 12-lead ECG, where you put a number of leads on your chest, and then they record electrical signals for each channel over the time. And then an experienced, well-trained clinician will try to interpret those signals to try to identify what's going on in your heart. So a typical standard, a schematic signal for one heartbeat is something like this. So you have a very big stroke here, which corresponds to your heart ventricle contraction, followed by a rather long refraction period. So you can see actually this interpretation process itself is rather subjective. And it provides very limited information, especially spatial information about your heart. So what we try to do is, what we call the next generation of ECG, is we want to directly, with the help of computer, goes into the heart to identify its conditions. For example, so nowadays we are able to build a potential maps on your body surface, and then we apply math techniques to inversely goes into the heart to recover its say electrical potential distributions. And so for example, this is the real heart potentials, how it varies for in the real world. So in order to achieve this, we need this so-called patient-specific modeling pipeline. So Rob has explained this basic pipeline to you. You start from images for each patient subject, you get very individualized data, and then you apply imaging processing techniques. And in order for computers to come in, we need to convert those imaging models into geometric models. So next generation is geometric processing. Upon that, you have a computer discrete model, and then we apply all mathematical modelings to describe this underlying physical process. And we get data, we visualize it, and we compare it with our experimental data, and this loop goes on and on. So, so in the next, I will just give an example of this pipeline for one case study, which is for the personalized treatment of cardiac defibrillation. The defibrillation is that the heart is not beating regularly, as shown here. This is a normal beat, and in defibrillation, your heart's going to be like this. So, in order to treat this defibrillation, we need so-called implanted cardiac defibrillator, which regularly sends electrical impulses to resynchronize your heartbeat. And so these are image scans of a human torso. You can see this is a battery, so this is electrically driven. And there's a long catheter goes through your vessel and into the heart. And then you can see there is a catheter lead here. So then it is going to periodically send electrical impulses. So the question here for clinicians is that what's the optimal locations to place those leads, and what's the correct amount of electrical impulses? It can't be too small, otherwise you cannot achieve your objective, and of course it can't be too large, otherwise you would feel very, very uncomfortable. So in order to evaluate these concerns, we first build up a patient-specific model from the images. So these are the real-time images of MRIs, and from these images, we do the so-called segmentation, which we label the different regions of different tissues and organs, and just isolate them from the background image. This process is called segmentation. So after segmentation, you can see all the region has been extracted. and after that, we enter this geometric modeling process. So actually here, so this skeleton structure is composed by all tetrahedral elements. So there are millions of tetrahedral elements which have a very high spatial accuracy. So if you zoom in, you can see those. These are all small triangles and dots. So now we have the geometric model. We can run computer simulations. So here with the model, you can see there's a small path, and here is the electrode leads, and the users can interactively change the position of the leads and run the simulation and immediately check its performance and effects. And we also can visualize the data. So these are electrical potential distributions resulting from your ICD interventions. And there are various ways for you to explore these data. So the clinicians may look at the left, which is your chemical density. So those red spots are the places near the near the leads where you have a very high electron density. And again, this is an evaluation of your data. And there are also other ways, more quantitative ways to get statistics of your interventions. And finally, so this is how actually it works in software. We just develop all our algorithm techniques into computer softwares, and this is called SCI, which is a basic pipeline-based software environment for solving scientific problems. And different modules play different functions, and you connect them to form the whole pipeline. You run the program, and then pops the window, and you can manipulate all the data. So that's it. And I would like to answer any of your questions.
What code type is that you're typing in? You just showed how it's like a pipeline interface, but what is what's your input type, I guess?
The input type would be data. Like say, so the input here would be we're reading some, say for example, imaging data, and also some measurement recording data, and so, of course, the first step of this pipeline is to try to combine those data from different sources to put them into the same model. We call this registration. So we can say, for example, for your image data, you get from MRI scans, and meanwhile, you have some electrical measurements placed on the body. And of course, you need to map those measurements into your imaging model, right? So that would be the first step.
What computer language are you using?
There are multiple languages. So the predominant language would be MATLAB and C++. So the basic idea is this. So all the very quick prototype developing and all your new ideas, new techniques are done in MATLAB in the first place. So just quick prototype your ideas. And as these ideas become mature, we then transform into, say, C++, which is is more for the engineering purposes, because C++ is more efficient and much faster. And also there are also a bunch of other programs, like say to build up these GUIs, we use TickleTK and Qt, and also there's JavaScript things and Python. So we have a very, very variety of computer languages.
All right, thanks, Stefan. I should say that the work that Stefan described certainly includes things that he did. Vidar is next. Is that right, Travis?
Yep.
All right. So Vidar is another grad student who works in this domain, works on a different disease, and we'll talk a little more about that disease and who wants to attend to this.
Now, leftover candy from Halloween. So we'll play a game. You can ask questions. If you ask a question, you get a candy. If I'm able to answer the question, I get a candy. So it's a win-win situation. So I'll use this straw at some point in time. As Rob mentioned, I'm Rob's PhD student. My undergrad was in electrical engineering. How many of you are planning to do electrical engineering? None? That's fine. It's not really a big deal. I did my master's in computer science. How many of you are planning to do computer science? Yeah, I changed over to bioengineering for my PhD, which is the field to be in anyways. So the past few presentations, you have heard about the heart and the electrical activity of the heart, and so on and so forth. So let me ask you, which is the biggest killer disease in the country today? Does anyone know? Heart disease. Good. Who answered that question? You did. Okay. That's taffy for you. All right. So can anyone tell me? Oh yeah, no categories. I'll make sure everyone gets one. Can anyone tell me where the heart is located? Left side, right side? Left side? Good. Okay. Here. Okay. Can anyone tell me what the function of the heart is? Exactly. So you were saying?
I said it pumps blood.
It pumps blood to the rest of the body. Very good. Now there is a trick to pumping blood. The idea is the heart generates electrical activity, electrical pulses, which I think Defang mentioned earlier. And that electrical pulses is used to generate chemical energy, which is what the heart uses to contract. And that contraction is what enables the blood to be pumped out to the rest of the body. And when the heart is functioning normally, the pulses happen at a regular interval, the heart beats at a regular interval, and the blood flows to the rest of the body. Every system is working fine, and you can go about your business without feeling anything. The problem occurs when things aren't going well. And one of the diseases when things aren't going well is called myocardial ischemia. Does anyone know what myocardial ischemia is? It's a free candy. Okay, that's okay. Yeah, that's okay. I'll show you a clip, 35, 40-second clip that gives you an overview of what the disease is. And then we can go from there. Assuming I can make it work. It's just a 45
Through
second.
the chest wall, exposing the lungs and pericardium, which is the fibrous covering of the heart. We can see the heart actually beating inside the chest cavity. There's the left coronary artery up close, and the right one as well. They supply blood to specific regions of the heart. Damage to or blockage in either of these vessels can injure the wall of the heart. If we focus in on the left coronary artery, you can see the red blood cells traveling through it unimpeded. But if the inner wall of the coronary artery becomes damaged, cholesterol plaque can build up, progressively narrowing the available pathway through which blood can flow. Clotted blood attempting to traverse the blood vessel may find it tortuous and too narrow for passage, and the artery may become completely constricted or blocked off. The blocked artery results in a lack of oxygen, or ischemia, to the part of the heart muscle that the artery supplies. The result is a heart attack.
Perfect. So, ischemia is essentially when your heart isn't receiving enough blood supply to maintain its normal functioning. And the first thing that starts going wrong is your electrical pulses start acting up. And then that starts affecting the heart's ability to contract, which means the blood isn't receiving, the blood is not getting pumped out, and then things start going from bad to worse, eventually ending up in a heart attack. So when a patient walks into a clinic saying, I have a chest pain, the first thing the cardiologist does is records the electrical activity of the heart, and that's what's called an ECG. And it has some squiggly features. I won't go into much details at this point here, but if you're interested, you can meet me offline, and I'll be more than happy to share what each of those features mean. And they look at particular features. The ST elevation, which is this isoelectric line, which is flat line in a normal person, but if someone's having an ischemia, it can be depressed, and if somebody's having a heart attack, it appears as an elevation. So that's the normal diagnosis for a clinician when the patient comes in and say, okay, you have ischemia or you have a heart attack. Now that seems pretty normal, shouldn't be a problem. But that actually is a problem because in clinical settings, there is 50% error rate in diagnosis. Meaning 50% of the time a patient walks in complaining of a chest pain, he might be diagnosed with something different. He might be just having acid reflux by having too much chili, but they might say, oh, you have a heart attack. Or it could be the other way around. So there are a lot of challenges in not being able to diagnose accurately. Some of it might be the way the recording is taken, the way where the electrode placements are. But we believe that there is a gap in our understanding of the electrical behavior of the heart, especially during the pathological condition of myocardial ischemia, which is what my area of research is. So how do I go about doing research in this disease field? Well, any research essentially goes through 3 major steps. You acquire data, you process data, and then you finally visualize and analyze data. Now, data acquisition, in my case, essentially entails doing animal model studies where we artificially induce ischemia by constricting one of the arteries and recording the electrical behavior by placing electrodes on the heart surface and also inside the heart. And once we have the electrical data recorded, we take the heart out and do an MRI scan. And you have seen some of those images from the other presentations, which something looks like this. You have the MRI scans. This is the left ventricle, that's the right ventricle. And you go through segmenting out those regions of interest by doing a little filtering, smoothing of the images, then identifying some areas that you are interested in, and finally generating a volume that might look like this. So that's what the heart segmented out from the MRI scans look like. And once we have that, we then register the electrical data that was recorded during the experiment onto this volume, and then we can then take slices through the heart surface. So let's say this is the volume, we have registered the electrical data, and then we can start taking slices and see what it looks like on each of those slices, and see if there's an ischemia, which is conventionally identified by a red color scale as electrical potential, and saying, oh, this is ischemia, this is where it is located, and this is how it progresses, and so on and so forth. So that's the basic process of acquiring data, processing data, visualizing, and analyzing data. But then the question becomes, okay, so what's the big deal? What's the big picture? So what's great about ski? And why am I spouting the advantages of working at ski? Well, at least in my field of ischemia research and people who do cardiac mapping, the way data visualization used to work was people used to manually draw those electrical potentials, sketch the heart, and manually create isopotential maps. And now, what we have today is the ability to not only generate a 3D volumetric model of the heart, but actually register that electrical potentials onto that 3D volume and then step through it through time. What happens at 10 seconds? What happens at 20 seconds? What happens a minute through? And not only that, you can actually cut through the heart surface through a cutting plane and see what the heart looks like. You can flip the heart over, see what the internal of the heart looks like electrically, and use that information to understand how the heart behaves electrically during a normal state and as it progresses into a pathological state. So the advantage of working at SCI, in addition to having one of the best coffee machines, is that we have world-class resources. We have access to some of the fastest computers, we have access to some of the smartest people, and you don't necessarily have to be the guy who has to know everything about everything, because the strength of SCI is collaboration. So although my focus is on actually physiological understanding of the heart, I don't necessarily have to know about how to visualize that data, writing code. I can look to Defan, I can look to Jess, I can look to some of the other people who focus on computational aspect and work with them to help me visualize that data. So that is the strength of the research that we do here. So for some of you who might be at some point be interested in following our path and going into doing graduate research, this is a great place to work at. For others who might plan to go back into industry, well, this is a nice, good to know information. And for some who just like to play Call of Duty, Modern Warfare 3, I've been there, done that. So at this point, that's the end of the, well, there's a moment of Zen. Well, one of the reasons the heart disease is one of the biggest killer diseases in this country is because of the growing epidemic of obesity. And this is like a fun slide, just to drive the point home. Who knows what the statue is? Michelangelo's David. And this was loaned to the US by France. And he happened to stay in the museum here somewhere for 2 years. And when he was returned to France, this is what he looked like. So, yeah, it's a big problem. So at this point, I'll be happy to answer any questions, hand out free candies. Please.
Segmentation is something that came up in all of the talks. Is that something that's done by a person sitting in front of
Yes.
saying this, I don't know, density of tissue is this certain thing? It's not animated.
Automated, you mean?
Automated, yeah.
What I do right now is manually because I know what I'm looking at. Now there are researchers in this facility who are trying to automate that process by trying to create a generalized segmentation and build an atlas. So that once you have enough segmentations of I look at it heart by heart, but if you had 100 hearts, you could probably say, okay, this is what the general segmentation looks like. Let's apply it on a new heart, because a heart might change its shape a little here and there, but in the end, the heart looks like a heart. So there might be minor variations here and there, which is what some of the researchers are trying to use for their benefit to automate that process. But at least as far as I'm concerned, I do it manually. One for you, one for me. Who else?
Where do you get your data from?
I have to actually conduct experiments. So we use animals, typically pigs, and then it's a typically a day-long process. I'm there at 7 in the morning, leave at 8 in the evening. And the preparation takes 4 hours, the actual experiment takes 4 hours, the cleanup afterwards takes 4 hours. So we are looking for some undergraduate students to help with the cleanup process. So. Okay. Are we out of time? I still have candies. Not for free.
I'll take a question.
Go ahead.
Have any of you taken like MRIs of yourself to friends or something?
There's one more student here who was supposed to give a presentation, but he was sick, so he couldn't come in. He had done a whole torso scan, the MRI, and apparently they failed to cover some of the parts. So when the scan came back, he was like, uh-oh. So because when you take a scan, they essentially take everything. So he had a problem. Yeah, so that was, to answer your question, yes. But it was just done for, I don't know why it was done. It's for a study.
So one of the questions that we didn't know the answer for beforehand was, how much does the heart move in the torso as a function of its own contraction? That's the part you probably know most about. How much does it move because of respiration? And how much does it move when you actually turn over? So it turns out if you talk to nurses who keep care of patients with heart disease in intensive care units, they will tell you that oftentimes in the middle of the night, of course, when you least want it, the alarms will go off on patients. They have monitors checking the ECG automatically on an ongoing basis. And all of a sudden those monitors will go off at night. And it turns out that one of the things that can set those monitors off is the patient turning over in bed. That's all it takes. So the question that they came to me with some years ago was, can you explain that by some electrophysiology or some physiology, or is it bad instrumentation? Is it the electrodes that are messed up here, or the instrumentation's bad? And it turns out that the heart will actually move far enough in the body through just changing orientation. So every time you roll over in bed at night, your heart actually flops a little bit inside the torso. And that flopping back and forth can change the ECG signals that are measured on the body. And those changes can be large enough to actually set the alarms on. And so we set out to answer this question of what's the expected variation in position of the heart inside the torso. And so obviously we had to get some data. Obviously a grad student had to go spend some time on an MRI map. Totally straightforward and understandable. And so yes, over the years we've had a number of students who've been imaged. And I too have been imaged. I've been scanned as part of studies before. So it's routinely part of the process. It's very educational too, of course, especially when you work in clinical research, to spend some time in that setting, to understand before you start sending patients, sort of into this procedure, to actually undergo it yourself and have a sense of what it's like, and what's easy, what's not so easy, what's so intimidating about it. Very helpful in this. So it looks like Tom has appeared on the scene just at the right time. So this is the final presentation we're going to do, and I showed you a couple of examples of this early, and this is software called ImageViz 3D. This is the software that visualizes enormous data sets on iPhones and iPads, as you'll see, as well as computers. And Tom Fogel here is actually not a grad student yet. He's going to become a grad student shortly. But he's one of our developers. So he's somebody who came out of graduate school in computer science and decided to move to a place like SCI in order to explore life as a developer in a multidisciplinary community like ours. And that motivated him not only to create great software, but then to go on and do a PhD. So, Tom.
Thanks. So I'm going to start by letting this go around because I tried to do this at the end. This is ImageViz 3D Mobile, and I'll talk about it towards the end, but you can just kind of play with it as I talk and go on. So yes, this is ImageViz 3D. It's kind of the main software project that I work on at SCI. And what it is a desktop volume rendering program for really large data. And so when I say desktop volume rendering program and large data, people tend to glaze over and not really understand what I'm talking about. So I'll go through and break that down and what that really means. So, oh geez, too many mice. There we go. So when we say volume renderer, what we mean is a program or a technique of taking a data set and looking inside it. So here you can see this Buckminster Fuller model that I'm rotating around here, and it's got some sort of green on the outside and some sort of red inside there. And what a volume renderer really allows us to do is decide exactly what we want to see in some type of data like this. So in this case, I already have a good what we call a transfer function set up, but let me just fiddle with it and make it awful so that you can see what happens when you change things. And so for example, if I do this, then I can see the outside of that structure very, very easily, right? And what that happens to do is kind of erase all of the other internal structure that we care about. So kind of the salient, nice characteristic about a volume renderer is that we can choose exactly what we want to see. And so we can see things inside the data set. And so I can get rid of this and see that internal structure. And so that can translate into something, oh geez, there we go. Like this data set here, when we're looking at a human scan or something, we take this scan, if we just visualize the outside structure, we're going to see all their skin and such, right? And many times, if you're a doctor, you're interested in what's going on internally with a patient. So you want to get rid of that external structure or make it just barely visible on the outside. And so a volume renderer really allows us to, we've had segmentation come up here many, many times, but volume renderer allows us to segment data in a very, very quick way, in a very, very coarse, I would say unrefined manner. And in some cases, this doesn't work at all, and you just can't do this approach. But in many cases, you can get a very good idea of what's in a data set by quickly looking through this transfer function and saying things like, I want to see what's on the outside, or I want to make that outside there, but just kind of barely visible, so I can still see that internal structure while I'm looking at the external structure. So that's ImageViz 3D, or a volume renderer, kind of in a nutshell, but what is large data, and why do we care about large data? And so volume rendering has existed actually since the year I was born. And we've had tons of volume renderers here at SCI developed over the years, and outside of SCI, it's a very popular rendering technique for three-dimensional data like this. But the issue that kept coming up, especially in recent years, is that we were getting these larger and larger data sets. And so we started getting these scans or these generated data sets that were 10 gigabytes or so, 20 gigabytes, which aren't huge by any stretch of the imagination. I mean, Rob was just talking about this 16.5 terabyte data set, right? But 10, 20 gigabytes is still something that you can't work with generally, right? You buy some new hot laptop or whatever, and it might have 4 gigabytes of memory. And so how do you fit that 10 gigabyte scan into that 4 gigabytes? And that's really what we developed ImageViz 3D to answer. And so if you take a scan like this, and let me turn on the bounding boxes so we can explain this. So this data set is a really fun data set for us because it's about 9 or 10 gigabytes, and I actually don't know how much memory is on this machine, but the nice part about ImageViz 3D is it doesn't matter, right? It doesn't matter if I have 2 gigabytes of memory versus 300 or something on a machine. I can still load up these arbitrarily large data sets. And the way we do that is via this bricking multi-resolution process. So you can see I can play with this data set, and even though it's 10 gigabytes or so, I rotate it around and things change very, very quickly with my mouse. And when I stop for a second, you'll start seeing that we get higher resolution data in there. And those green boxes actually represent how much data we're loading. And so each one of those little chunks is a constant size in memory. And so we can render one of those chunks and then take that and throw it away. And by doing that, we can render another little chunk in there. And if we keep iterating this process, we can really render an arbitrarily sized data set, using any small amount of memory. And so this has really been an amazing tool to let people visualize data that they really couldn't see before. I actually just got a 7 terabyte data set from some angiogenesis collaborators over in MEB, and they gave me this data set and said, what can you do with it? And one of the funnest parts about my job actually is taking these 70 terabyte data sets and then generating an image or some movie or something and give it back to them, because everyone always comes back and goes, wow, I've never been able to see that before. It's like the first time they've ever been able to visualize their data ever. And so it's a really powerful tool. It's a really rewarding process there. So that's one data set there. Another data set I wanted to go over here is this simulation of a hydrogen flame here. This is actually done on another supercomputer at Sandia National Laboratories. And the fun part about this is what's going on inside the data set. And so there's these fluids that they set up the simulation with, and then they start this burn process over on the right here. And they start seeing, how does this flame evolve over time, and how does it generate things, and generate new flames, and flames die out. And so what they really care about is what they call these extinction regions. And so these extinction regions are kind of these small areas within the data set where flames sprout up or where they come down very quickly. And what they want to happen is for those extinction regions to sprout up right at the start and just kind of stay at a constant burning flame. They don't want it to sprout up and go really bright for a second and then come back down and die. They want it to just stay up and burn continuously, because this means they've generated a good fuel. They've decided which mixture is good to generate a clean burning constant fuel. And so a new rendering, oh geez, which one of these? Okay, good. A new rendering technique we developed is something called Clearview, which really allows you to do that quite effectively. Let's scale this back a little. So the fun part about Clearview is that it's very, very simple here. You can see I changed this little slider here, and I changed this little slider here, and we see different parts of the data set, right? And so what they're really looking for is, there we go, these tiny little things here. So this tiny little thing that's sprouting up here, or this tiny little sprouting up here, but they also want to see what's going on in the overall data set. And so what Clearview allows us to do is say, let's focus on this particular region here, but I also want to see some context information of what's going on the outside. So you can still see this kind of curvature that's going on, the general flame as well as that extinction region. So that's most of ImageViz. I guess the last thing I want to show you is 2D transfer functions. Go away, data set. Let's do it with this data set. Maybe I have the backpack actually. Let me just set something up real quick so we can back. Oh, I don't have backpack here. Sad face. Okay, we can do it with this data set. So what happens when we have this one-dimensional transfer function? You can see this is very complicated, and that can be very difficult to differentiate different regions of interest. In particular, if you have something that's changing very rapidly, lots of times you want to differentiate that based on the area that changes versus the area that's kind of uniform and contiguous. So in this data set, great, let me rotate it a little bit so you can see it. In this data set, you can see that this blue area as well as the red area that I can't quite reach is largely contiguous, and then you have this area where these fluids are mixing that is much more happening. There's much more going on there. And so in our 2D transfer function editor, we're not just looking at data values, we're also looking at the changes in data values. And so along our x-axis here, we have our data. And so this is the zeros of our data set, and this is, I don't know, maybe the thousands in our data set or so. Along our y-axis, we have a new aspect of it. We have the gradient. And so we can differentiate between things that are changing rapidly versus things that aren't changing so much. And so we can see if I find that one quadrilateral, there we go. If I take this and move it, oh, that's the wrong keyboard, that's why. And move it way down, we get just that homogeneous region at the top there. But if I move it way up and I get this, then what we get is the transition region there. And that's where the region where everything is changing. And so this gives us a powerful new tool for looking at, not just those homogeneous regions, but actually differentiating them just based purely on those data values. So that's about the extent of Images 3D. Are there any questions about not just Images, but really any of the software we worked on here? I tend to get my hands dirty with all of them, so I'd gladly take them now. Anyone, Bueller? Yeah, that's my problem.
I had a question.
Sure.
I think you might have explained it, I was playing with that. So how do you get like a terabyte to show up visually? Is it just feeding the image and it's not actually manipulating the data? How do you do that?
So really we're just loading things up in little tiny chunks. I think I can close everything. So we have these green boxes that you can see. You can see if I maximize it probably. And we only need to load up one of those green boxes at a time, right? So we can load up just the chunk here and just the chunk here. And we can render one of those chunks, and then when we're done with that, we throw it away. And then that gives space for more of those chunks. And so we can just kind of keep doing that, and we can load up infinitely sized data. To an extent, we're also a little smart about this in that, if you have a terabyte of data, and you have a screen size that's 1900 by 1200 or something, you can't see a terabyte of data. It doesn't happen that way. You need a much better display to actually see that. And so we'll actually figure that out and say, well, it's not worth rendering a terabyte of data. Let's render 10 gigabytes instead, because you won't see the difference anyway.
Okay. So in that case, you would just sample every 10 data points you actually render?
Sort of. We build what's called a Gaussian period.
Okay.
Yeah.
Sounds fine.
It's just a way of subsampling the data. One way of subsampling is just to ignore every other data point, for example. And another way is like a Gaussian pyramid, where we average out the points that are neighboring. And so you just generate that in a hierarchy, and you can choose which level of that hierarchy is most appropriate given your current viewpoint.
So is it like streaming? So it's stored somewhere, you create the image, you produce the image, and if you want to change something, it streams new information.
Right, exactly. So when I start rotating, for example, you see, or zooming in, I guess, you see that this is a very low resolution, right? You can barely see what's going on here. You can see, well, there's a little tiny heart, there's some, big vascular structure there, there's some bones, but you really can't understand. And it takes a sec to actually load in that higher res data. And so we're loading in that coarse data so that we can give some interaction, but then as you stop, it says, okay, I'm going to load up one of those chunks and render it. And then it throws that away and loads up another one of those chunks. And it just so happens that it can load up, about 5 of those chunks per second. So sometimes you can't really see that it takes a sec to render all of those, but it is doing that. Any other questions? All right, well, thanks, guys.
I think that's everybody we had lined up for today to talk to you, but if you have more general questions, feel free to grab any of us now on your way out. And like I say, if you're curious at all, go to the website. You can find lots of connections there to images like this. Shems keeps an amazing array of videos on the website. You can download some of those and have a look at them and get a sense of some of the projects going on here and some of the people behind those projects. And if you are interested, feel free to contact individual faculty members just send them an email, explain who you are, what you're looking for, that you're interested in involvement in research, and like I say, keeping persistent. Any other questions before we head off? Okay, thank you all, and have a great rest of the day.
What exactly were we doing at this lab? What's like an average, what's an average day?
I don't know if there is one.